on analysis of label-free quantitative mass spectrometry data for E. coli, and it's presented by Cormac Monaghan.
Okay, can everyone see?
Yeah.
Okay, good. Wonderful. So yeah, so thank you, Connor. So my name's Cormac, and I'll be walking through a group project that I was a part of last semester, which is an analysis of label-free quantitative mass spectrometry data for Escherichia coli, or as most commonly known, E. coli. So while it's just me presenting today, this work was a collaborative effort, and I would like to thank both my teammates, Beatrice and Andre, and also all the supervisors listed below. But so starting off, I was going to walk you through an outline of what the presentation would be. So for the first few slides, we'll start off with the project background, and then walk you through how we processed and analyzed the data that we had, and finally sum everything up and present some future work. So starting off the project background, recently there was a paper published which compared the proteomics of the multi-drug resistant plasmid containing E. coli that was grown in several different antimicrobials. So that's a lot of big words right there. So let's break them down into more simpler terms. So proteomics is simply the identification and quantification of what is known as the proteome, and in our case, that is the proteins produced by bacteria. Plasmid is a small bit of circular DNA that is separate from the bacterial DNA itself and is often linked to resistance. An antimicrobial is a medicine used to treat and prevent infection, and in the case of bacteria, they are antibiotics. And then multi-drug resistance is simply a lack of susceptibility to any of those antimicrobials. So back to the paper that was published. So these E. coli samples were grown in addition with 4 different antibiotics, them being ampicillin, cefotaxime, imipenem, and ciprofloxacin, and then an E. coli sample on its own, which was just the control sample. So once these bacterial cultures were grown, they were sequenced, and the protein data was analyzed, and the paper was published. And then where do we come in? So using the data from this study, our overall aim was to implement a more robust statistical approach compared to the current standard, which is a software known as MaxQuant, to analyze label-free proteomic data, especially with small sample sizes. And a secondary aim was to ascertain biological meaning behind our results in the hopes of finding interesting proteins related to antimicrobial resistance. It should be noted that data like this is often difficult and expensive to obtain. External labs are often needed. The machinery used is both expensive to purchase and also expensive to run. And even as an end result, you can end up with data with lots of missing values and also quite small sample sizes. So in our case, we had 4 samples, but we only had 3 replicates per samples, which is less than ideal. So having a robust statistical method is imperative as allows you to retain as much data as possible and also give you more confidence in your end results. So then for the next few slides, I'll walk you through the processing steps that we took. So for a quick overview, we start off with the raw intensity data, so the raw unprocessed data obtained from MaxQuant. And we integrated that data into an R package known as QFeatures, which is just an infrastructure package that let us to store everything in one object and assign a hierarchy as opposed to having everything in separate data files and everything being messy. We then process the data using 4 fundamental processing steps. So aggregation, filtration, imputation, and normalization. Once the data was processed, we can analyze the protein abundance levels to see what effect each antibiotic has on the E. coli. And finally, we can look at our results from a biological perspective and ask ourselves if they make sense. So start off with the processing. So like I said, there's 4 standard steps, and I will go into more detail about each of them, but just for a general overview, we used the median to aggregate from back down to the protein level. We adopted 2 filtration steps to filter out unreliably measured proteins. we also use K and N to imputate missing values, a little bit deja vu there. And we use fastest cyclic lowest normalization to normalize our data and reduce technical variability. So we'll start off with aggregation. So our first question obviously is, what is aggregation? Well, since we're analyzing proteins, we obviously want a data set filled with protein values. But because the way the E. coli samples are sequenced, they get broken down into peptides, which, if anyone remembers biology, peptides are the building blocks of proteins. But we can use these peptide values to get back to the protein level because we know which peptides are assigned to each protein. So in this example here, these seven peptides all correspond to a single protein. So we can use measured values to get back to the protein level. So typically, research teams would sum up these values, and that's what the previous research team did. However, we propose that using the median holds several advantages. But right from the off bat, you can see that depending on what aggregation technique you use, the results you obtain are vastly different. And similarly, your overall distribution of results changes greatly. So like I said, summing the values in each level is a common approach. However, you can see that this technique does provide a wider, more diffused distribution, whereas using the median gives you a more narrow systemic distribution. And this difference in distributions kind of has a great effect on your end results, so which we will see later on in the presentation. But you might remember me mentioning that the median has several benefits over the sum or the mean. So the median is classified as what's known as a robust statistic. It's able to hold up to less than ideal conditions, which is what we have in this case. So it's less susceptible to data contamination and has what's known as a 50% breakdown point, which is the maximum percentage of observations that can be replaced with outliers, missing values, extreme values before causing an unbound change in the estimate. So to better illustrate, say we had the numbers 5, 10, 15, 20, and 25, the median would be 15. But if we were to change the last value to something extreme, like 1,000, the median would still be 15 because it's still the middle value in the sequence. But if we were to sum up these values, our end results would obviously be greatly different. But we may be mentioning that we have less than ideal conditions at the moment. So missing values are quite common in this line of work, and we do have quite a few in our data. And that's particularly due to low signal machine detection. So at the peptide level, 57% of our data is missing. Now, aggregating does help. Once we aggregate our data, only 23% of it's missing, but we still need a way to address these missing values. So that's where our next processing step comes in, which is filtration. So, like I said, we adopted 2 filtration criteria to help filter out unreliably measured proteins. The first being proteins that have less than 4 peptide matches. So if you remember my previous example of the 7 peptides corresponding to a single protein, well, if that protein had less than 4 peptides, it was filtered out of our results. So that accounted for 25% of what we currently, the data we currently have. And the second step was proteins that have more than 50% missing values across all their samples. So that accounted for a further 21%. So at the end of both steps, we ended up with a data set of 1,071 proteins. Now, these proteins still have missing values in them, and we need to deal with them. So that's where the third step comes in, which is imputation. So a traditional imputation approach is to focus on the lower tail end of distribution and imputate from there. So in this case, the left end of the distribution. However, because you're focusing on the lower tail end, you can often lead to an inflation in your variance. So that's why we followed a recommendation from a recent paper by Lazar in 2016, wherein the team compared various imputation methods and showed that the K&N approach was one of the best performing methods. So you can see from the distributions in blue, so blue-green, that most of our imputed values are almost more centralized. They're just less than the mode or the median distribution, which helps lower the inflation in our variance. Then moving on to the last processing step, which is normalization. So normalization aims to remove any systematic differences introduced during the experimental process. So typically introduced by the mass spectrometry machine. And it shows that any differences between our samples is driven by biological and not technical bias. So in the original paper, the authors used what's known as the Z-score method to normalize the data. However, this method doesn't provide a sustained reduction in the variance between each of the replicates. You can see the distributions in red in the graph, they're still quite wide and dispersed. So again, following recommendations from current literature, we use what's known as fast cyclic lowest normalization. And you can see that when you compare both the methods, the Z-score method gives a more dispersed distribution, whereas the fast cyclic lowest method gives a more symmetric distribution. So the alternative method is more centralized around the 25 mark on the x-axis. So once we've processed the data, we can move on to what's known as a downstream analysis. So we can further analyze the data and draw conclusions. So this is broken up into two steps, a protein abundance analysis and biological annotation. So starting off with the first one, protein abundance analysis aims to identify proteins that have significantly different abundance levels given a specific treatment when compared against the control. So we want to analyze we want to compare each protein in a certain treatment against the same protein in that control. But you might remember that we have 1,071 proteins and we also have 4 samples. So that's over 4,000 multiple comparison tests that need to be performed, which is quite normal in the field. But a common multiple comparison test that is used is a student t-test, which does have several drawbacks. The main one being its high false discovery rates. So that's why we proposed using what's known as a moderated t-statistic, which was introduced by Smith in 2004 and is available in this limit package R. So without going into too much detail about the moderated t-statistic, it kind of calculates a more robust estimation of the variance by sharing information between all the proteins and by using some empirical Bayesian arguments. So we do have a technical report if anyone is more interested and would like to know more, which is linked here. And there's also a QR code at the end of the presentation that links to a GitHub repository. So, but once all of these tests were performed, we needed a way to visualize our results, which can be done with what's known with a formal scatter plot known as a volcano plot, which if you concentrate and use a little imagination, kind of looks like a volcano. So on the x-axis, we have the fold change, which is simply the means of the treatment for that protein divided by the means of control for the same protein, and then put into the log2 scale. On the y-axis, we have the minus log 10 p-value. So each point on this graph is a protein, and we label the top 10 significant proteins per group in red that met like certain significant thresholds. So those thresholds being a p-value less than 0.005 and an absolute log fold change greater than 0.05, which is the current best practice in the literature. And overall, this approach allowed us to classify proteins that are both different in biological terms, which is the log fold change, and statistical terms, which is the p-value. So here we have our results for our ampicillin, cefotaxime, and then imipenem and ciprofloxacin. So, but what if we compared how our method performed to the original method? Well, if we applied both the significant thresholds, we can see that our approach that in most cases detected significantly less abundant proteins. So you can see that for ampicillin, ciprofloxacin, and imipenem, we detected less abundant proteins, the exception being cefotaxime, wherein we can highlighted one extra abundant protein. But to delve a little deeper, we can ask ourselves, well, are any of the proteins we found similar to the original paper? And for ampicillin and imipenem, we didn't. There was no overlapping proteins. However, for cefotaxime, 2 of the proteins were the same, and for ciprofloxacin, 4 of them were the same. So we can then go a little deeper and ask, well, what about the most abundant proteins? And you can see from each of the graphs that, so if you look at the bars in blue, in most cases, in these cases, these are the most abundant proteins that we found that were significant. But if we compare them to the bars in red in the original paper, these proteins weren't actually, in most cases, were not actually found to be significant. So our method actually highlighted new proteins that were classed as significant compared to the original paper. And similarly, in some cases, our paper highlighted proteins that were actually not found in the paper, in the original study at all. So it's quite interesting, but it does lead us into the second part of kind of our aim, which was the biological annotation. So we had to ask ourselves if the results that we found made sense from a biological perspective. So we can use what's known as the string database, which allows us to input the protein names and then get outputted the functions of each of these proteins. However, these functions are in what's known as long text, which is it's a detailed list of each of the functions, which is very useful, but also did lead to some issues further down the line, which I will go into. But if we were to look at the top 3 proteins that we found, and just for sake of time and easiness, I'm only going to focus on the first box, so the red ampicillin box. So in the original paper, the top 3 proteins were more so to do with translation and plasmid transference, while in our approach, like our analysis, the proteins that we found that were most abundant were more so to do with oxidative stress, which does make sense because if you grow a bacterial sample with the addition of an antibiotic, it's obviously going to cause stress on that bacteria. So that's good. So our results do make sense. And then if we again look at the common proteins, well, for cefotaxime, we noticed a low relative abundance in the catabolism of glycans, which again does make sense because putting stress on the bacteria gives the bacteria less energy, so then has less energy to catabolize. However, one interesting result that we did find that we couldn't really explain was for ciprofloxacin. So if we look down at the very last part, we can see that we have a lower relative abundance in ABC transporter. However, the ciprofloxacin antibiotic plays a role in the DNA of the bacteria, which an ABC transporter is related to. So it did make sense to us to have a lower abundance of that, but it was an interesting result which did warrant further investigation. But so for a future direction of the study was, you might imagine me mentioning that the functions of each proteins are in long text, which is detailed, but it also means that there is a load of unnecessary words in the functions. So if we were to use a word cloud to visualize what are the most important functions of each protein, we often end up with words that don't need to be there. So if we look at this example here on the right for ciprofloxacin, we see that we have words like from, cycle, factor, which don't need to be there. So a typical way to deal with these words is to use what's known as a stop word list. So once this word is detected, it's removed from the analysis. However, at the moment, a stop word list for proteomics doesn't exist. And you need quite good SQL skills to implement one, which we didn't have at the time, and we're also quite short on time at the end of the project. So future direction of this study could be developing and implementing a stop word list to further analyze and process this data. But to sum everything up, we proposed a new, robust, open source method of analysis using R and various bioconductor packages, and moving away from the current max quant software. So overall, this allowed for greater transparency and reproducibility in our results, which are both kind of fundamental for analyzing data like this. However, and although some of our results are similar to that of the original study, our approach does highlight new proteins which may warrant deeper biological investigation. However, I do want to note that we're not arguing that our approach should be adopted as the current standard, because due to our unique processing approach, the results we contain are not overly comparable with that of other studies. So we would need more studies to use this approach so we can properly compare everything. However, it does raise the question of if alternative workflows should be used to analyze mass spectrometry data, as you can see that depending on what method of analysis you use, you end up with vastly different results for your end product, but for your end results. So there's our references, and if anyone is interested, I have a QR code for a GitHub repository which has plenty of reports and more details. So thank you.
That was a great talk, Cormac. I just wanted to ask there now, does anyone have any questions for Cormac? Oh yeah, Aoife has a question there,
yeah. So that was very interesting. I was just wondering, did you run any correlation matrices or clustering analysis?
Yeah, we didn't, but at the start of a project it was one of our overall aims. But you might remember mentioning that. One second. Go back. Yeah, so we only have 3 samples in each of our groups, which did cause the issue that for correlation or clustering, you need at least more than 3 samples, since one of the equations in your denominator is n minus 3. So n in this case would be 3. We'd have 3 minus 3, which would be divided by 0, so which would just give an undefined number. So at the time we couldn't do it, but if we did end up having more samples for each of our groups, it was definitely something we would like to investigate more. Hope that answers your question.
Thank you.
Do we have any more questions for Cormac there at the moment? But I have another one there, Cormac. I was wondering, why did you choose your filtration steps?
Yeah, so yeah, kind of it's, yeah, here we go. So kind of common practice in the literature at the moment is if you have kind of less than 4 peptide matches for your proteins, it's once you start aggregating back to the protein level, oftentimes you end up with an unreliably measured protein. So that's why you filter out with less than 4 peptides. And similarly, it's the same with having more than 50% missing values across all samples. Just kind of end up with kind of an unreliable protein, which may affect your results at the end. So it's best practice just to filter them out during the processing and leaving the rest of the reliably measured proteins.
That's perfect, Cormac. Thank you very much.
Thank you.
I'd just like to say thank you, Cormac, for your presentation. It was a great presentation, and I guess we can move on to the next presentation, which is learned image compression, a comparison between JPEG and neural network methods by Paddy Gorey.
Okay, great. Thank you, Connor. I'll just share my screen. Okay, hi everybody. Thank you for hopping onto the call to watch our presentations. So as Connor said, the presentation I'm going to be going through today is learned image compression, a comparison between JPEG and neural network methods. Before I start, I'd just like to thank my colleague Malat, who I worked on this project with during the Winter Symposium for our first semester group project. And of course, our supervisors, Tom Naughton, Roseanne Deo, John McDonald, and Charles Markham. So myself and Malat worked on 2 sort of distinct methods. We kind of each focused on one. So Malat's giving me some notes on her side of things. So I can hopefully do justice what she's done. But anyway, we're going to start off with, first off, what the aim of this project was, and that was to create and analyze a couple of learning-based image compression pipelines. We've seen a lot, just in general, in tech and statistics and machine learning, of using data-driven methods, or different machine learning or AI methods to apply to long-standing problems or long-standing sort of issues in computers and in technology. And this is the sort of idea behind what we're doing. We're getting a learning-based or a data-driven method to image compression and see how it goes. So we want to compare the performance of these pipelines with the JPEG standard. Now, what is the JPEG standard? Well, hinted by the very pixelated cat images, and I can assure you it's not the first image of an animal you'll see throughout this presentation. Let's talk about what image compression is and what JPEG is. So JPEG is a form of image compression. It's sort of the standard, one of the most ubiquitous image compression methods around. And image compression can come in two forms. So there's lossless, so that's compressing an image. So you're reducing the size of that image, but you're maintaining the image quality. So an example of a lossless image compression codec would be PNG. That'd be one of the most common. And then lossy, which is sort of the focus of our project, and that's what JPEG is sort of best known for, is lossy image compression. So that's compressing, reducing the size of that image, but you're also reducing the image quality. So it ends up being a sort of balance between reducing that file size while not reducing the quality too much that it becomes a bit unusable. So just an example here that I'll go through in a bit more detail with these cats again. But just a quick nod to JPEG. So Joint Photographic Experts Group is what JPEG actually stands for. And that's just, again, the kind of de facto standard for image compression, established in 1992. And it's based on the discrete cosine transform. But I'm not going to get into the weeds on that, because that's not what we're really here to do. So just to kind of zoom in on this example of lossy image compression. So we have this image of a cat lying on its side, and how JPEG works is it uses a metric called quality. Now, quality, you would think, would be something like, if I set it to 100, it's going to be 100% quality. If I set it to 50, it'll be 50%. It's not really quite how it works. It's sort of a measureless or a unitless sort of measure. So, for example, I set the quality in compressing this image with JPEG to quality of 5, and that gives a compression ratio, which is the ratio of the original file size to the compressed file size, a 16:1. Now, you would think maybe a quality of 5 would be 5% of the original size, but that's not really the case here. So it doesn't quite track. But what you can say is that a low quality is going to produce a very small image. So we can see the sort of artifacting or pixelation that can occur with JPEG when it compresses image. Just zooming in on the same section of both of these images, you can see there's some pixelation here on the eye, and you can just see that's just sort of how the DCT works. It works in grids, essentially 8x8 grids, so you end up naturally getting this sort of pixelation. So JPEG is capable of high compression ratios with minimal distortion. And that's why it's so ubiquitous and so widely used and so effective. So now we ask, well, where do neural networks come in? So just looking at the sort of big picture of this project, there's 3 methods that were looked at. We've quickly gone through what JPEG is, and we have 2 machine learning or learning-based methods then that I'm gonna walk you through. So just a quick run through again of JPEG. So we can see sort of we have the 3 pipelines on the same sort of footing, so we can compare similar steps that they have. So just starting with JPEG, we have some pre-processing with the DCT. There's some lossy compression and lossless compression with quantization and some LZ77 and Huffman. We end up then with our compressed image, which in this case is just a JPEG file, so .jpg or .jpeg or whatever. Then in the end, there's some lossless decompression with some entropy decoder, with some entropy decoding, and some lossy decompression with some rescaling, and a special sort of post-processing step with JPEG is the inverse DCT. But as I said, we're not going to go through JPEG in too much detail. So we're going to move to the first pipeline, which is the ESR-GAN, which is the first of our learning-based methods. So first, what is a GAN? So a GAN is a generative adversarial network, or one of many different popular neural network architectures that can be used for a lot of different things. So a GAN consists of two neural networks that are sort of trained simultaneously in a kind of a competition. So you have the generator that learns to create fake data by incorporating feedback from the discriminator, which essentially is a classifier that determines if input samples are real or fake. So basically you get the purpose here is to learn which of the data that's being put in is real or fake, and the discriminator essentially makes a prediction between 0 and 1, where real data is 1, fake data is 0, and then there's a sort of feedback loop here between the generator and the discriminator, and the generator will learn to be able to produce this data at the end. So how or why do we want to use a GAN? Well, we want to apply super resolution. Now, super resolution is a method essentially of getting a low resolution image and producing a high resolution image. So rather than just say getting a low res image, putting it into paint and dragging it and scaling it up, you want to actually scale that up, but have it actually be a bit more aware of what's going on, what's happening in the image, and have it actually scale it up so that it looks like it's a high resolution image, as the example of this mandrill image here. So it tries to reconstruct the original scene with a higher resolution, given a set of images that are at a lower resolution. So it essentially, if you're going to use a neural network method for super resolution, you're almost sort of teaching it how to, sort of see what's important in scaling these low resolution images up to these high resolution images. So a quick look at the sort of pipeline of how the GAN works. So we start off with a high resolution PNG file. So with another way of thinking of this GAN is that we'll see later with one of our different networks where we train the encoder and we train the decoder. In this case, you can think of, we're not actually training the decoder. So the thing that actually compresses the image isn't an actual learning base for a neural network, but the thing that is decoding that, so actually getting your compressed image and uncompressing it, is a neural network based method. So essentially we have an original PNG file at high res. So this is resized, so it's downsampled, depending on how much compression you want. So you can compress that, you can downsample that to 5%, anywhere between 5% or 100% of the original image. So you're going to get a PNG that's going to have some sort of loss because you've scaled it down so much. And then what we do is, so that image then is our compressed image basically. And then if we want to bring that back up to the original size, then we can use our super resolution to upsample that image and get hopefully what will be an exact recreation of our original high-res PNG file. So just a quick example of what we mean by this. So we have this original cat on the left here. It's downsampled to about 10% of the original size. And we can see that's very pixelated here, or rather very blurry. And then that gets upsampled to the same original resolution as the original PNG file. So we see that there is some pixelation in that occurring here. In the same way that for JPEG, if we were to comparatively compress it down, we see that the file size here are the same. So in the ESR GAN, our compressed file is about 8.7 kilobytes. And the equivalent would be setting the quality to 0 in JPEG. This was just done through sort of trial and error, because like I said earlier, the quality metric doesn't really have a direct correlation between, say, a percentage or things like that. So we can see that these images are both sort of compressed the same sort of amount, and we can see the distortion here, but I'll get into that in a little bit more detail. So now we're going to look at what my main focus on for this, which is what we call the CAE. So this is the compressive autoencoder. Now, what an autoencoder is it consists of two different neural networks, again, trained together during the training process. So there's an encoder and a decoder. And in the middle here, we have our compressed file. So what we do with this is we have our input image goes into the encoder encodes that into a file format. And then in a practical situation, if you want to store all these images, but store them in a compressed manner, you just store those files. And then whenever you decide that you need to get the original image and reconstruct it, you feed that through the decoder, and that gives you your reconstructed image. So the encoder network essentially learns an efficient manner of compressing the image. So you can think of the encoder as sort of, so it uses a convolutional neural network to do this. You can think of it as taking in images and almost sort of looking at what the most important aspects of it, say different layers in that network are going to pull out different features that are important. For example, lines or changes in color or things like that. So the encoder is looking to learn the best way to essentially create this small representation or latent representation of the original image. And the decoder then is just looking to reconstruct the original image from that latent image in the middle. So the aim then is that the input image and the reconstructed image should be identical, as they should be as close to each other as possible. Now we'll see for our implementation of this, that's not the case, but that's the ideal that we're sort of striving for. So just to say how we actually represent these images. So with JPEG, the compressed image is simply just a .jpeg file. But for the CAE, the actual compressed file is usually we store it as a NumPy file, and then it's all put into a zip file. But we use this quantized representation where we essentially say we have one image. This is about 512 digits. So this is one number that's 512 digits long, and it consists of ones, zeros, and twos. So this is a base 3 representation. So by using this sort of quantized representation, just using base 3, it encourages the network to learn a more efficient manner of compressing, because usually in autoencoders, that latent representation, depending on what the autoencoder is doing, it's usually going to be a tensor of some size, or basically a 4-dimensional array or multi-dimensional array of a certain size, and each of those values is usually a float number, a floating point number, which is be about 32 bits, a 32-bit floating point number. So if you had 512 of those, well, the space that you actually end up using to store this information ends up sort of getting away from yourself. Whereas if you have this much, much smaller representation, you just have one number that's 512 digits long, again, it's training it to look for a more efficient way of actually compressing that image. So just to show an example of how CAE works, we have another cat. So CAE works with images in 32 by 32 patches. So this image is cut up into 32 by 32 patches. The patches are all fed through CAE, and the reconstructions then come up in the end. So that's why we have this sort of grid-like pattern appearing in our reconstructions. So the CAE anyway is trained with the Linnaeus 32 image classification dataset, which is just an image classification dataset with various categories of natural images and buildings and cars and things like that. So here we get a compression ratio of 3.5 to 1. The JPEG example for your memory earlier on was getting about 16 to 1. So that's a sign that this isn't performing as well as we would hope. We'll get into that more in the results. And the distortion here is 0.0424, but again, we'll get into that in a moment. And just to draw your eye to it, in case it's a bit hard to see on your screen, of just the grid-like structure that kind of appears when we reconstruct our images. So just to quickly talk about the methodology for how we actually compare these methods. So the rate distortion curve is a standard in image compression in terms of determining how well it does. So rate distortion, the rate, which is how much the image is actually compressed. In this case, we use compression ratio, which is the ratio between the original size and the compressed size. And then the distortion is the difference between the original image and the reconstructed image, the uncompressed image and the reconstructed image. So like I said, with CAE, the aim is that the original and reconstructed image are the exact same, but that isn't usually the case for our methods here. And distortion is measured using the mean squared error, or MSE, as I'll be referring to for the rest of it, which is essentially is calculating the Euclidean distance between pixels. So it's just getting your two images, going pixel by pixel, and just calculating the difference between them. So, if they're different colors or anything like that. So it's a straightforward metric, one that would be probably better suited for image compression or comparing images, is the structural similarity index metric, which basically takes into account more so, in an image, things near each other are probably going to be related, and there's going to be an inherent structure there, whereas MSE is just looking pixel by pixel, what's the distance between them. But the MSE is a bit easier to implement for this. So the lower the MSE, the closer the reconstruction is to the original. So like I said earlier, image compression is all about striking a balance between compression and distortion. So just a quick mention of the images that we use. So we used a subset of the OST image data set, which is outdoor scenes, train and test. So the resultant distortions and compression ratios are calculated for each image in each pipeline. We can see different categories here: building, sky, plant, animal, and so on. And we essentially feed these images into these three pipelines. We get rate distortion curves for each of the pipelines, and that's how we'll compare them then. So just to quickly jump through the GAN, this is just an example of one single image that's gone through at various compression ratios for the GAN and for the JPEG. And we can see here the GAN and JPEG are sort of on, in terms of their mean squared error and compression, in terms of their mean squared error, so their distortion, on relatively even footing. We see that they're both reaching about 0.08 at sort of highest compression ratios. But what we're actually seeing here is, as well, if we look at our plots, look at our axes, is that the compression ratio for the GAN, we're getting up at above 200 compression ratio, which is quite an extreme value compared to JPEG, which is only getting about just over 50. So we'll get into this a little bit more later, but maybe that's a hint that there's some cases where the GAN can perform better than JPEG. So this plot is the same as the previous one, except now it's 20 different images rather than just one. So we can sort of see, the similar behavior for each of the images. We can see that, as is the case, JPEG maybe a bit less so, depending on what image is being compressed. The rates sort of fluctuate a bit more. That's a bit more noticeable in the GAN. So this image on the bottom here is being compressed, or it's getting a much lower distortion compared to some of these higher images. But that can just be the nature of what's in the image. Maybe it has different colors or different sort of shapes or large open backgrounds or things like that. So these methods can kind of be sensitive to what you're actually training with, which is something that I'll get into with the CAE in a couple moments. And this is just to highlight ignoring the different images, just looking at GAN versus JPEG. We can see that there are a lot of cases. Now, saying that overall here on this left side region, where the sort of general compression ratios you get with JPEG, for the most part, JPEG is getting lower distortions. But the further we get up in terms of compression ratio, we can see that there are cases here where the ESR GAN is actually getting lower distortions compared to JPEG. And over here in this region of very high extreme compression ratios where JPEG doesn't even touch, is interesting, just because it shows that JPEG is, maybe better for a sort of standard generalized compression. But if you're looking for really extreme compression ratios, over 100, 150, we can see that the GAN is sort of capable of that, whereas JPEG isn't really. And this is just another quick example using the images that we had earlier on of just showing where they lie on the graph. We see our original image downsampled to 5%, and we can see the varying levels of distortion here. So we can see that the distortion measure is about the same, but at the same time, we can see that this image from the GAN does seem a lot more pixelated compared to the JPEG. But as well, this is a very extreme compression ratio about 250 compared to a compression ratio of about 50. So it's hard to compare, such an extreme compression ratio to a much smaller one. So now just to quickly go through the results for the CAE. So CAE doesn't perform as well as the GAN, unfortunately. So this is a plot similar to that shown in the ESR GAN section. We see various images compressed at different compression ratios with JPEG. And we see that it has a fairly regular behavior of its as it's compressed more. We get increasing distortion, but generally it's staying to very reasonable values. Now, over here on the left is the CAE. So we see that it's getting, if you zoom into each of the lines, an individual line, it's getting similar behavior, but overall it is a bit more varied. So we see that, for one thing, CAE is, as I mentioned earlier, is very sensitive here to what image is actually being compressed. So we see the difference between the bottom blue image and the top purple image there is quite large in terms of their levels of distortion. So one is being compressed with a lot more distortion, while one is being compressed with a lot less rather. And as well, just in terms of comparing CAE and JPEG, Whereas GAN was capable of some extreme compression ratios that JPEG wasn't. CAE, or this implementation of CAE, has the opposite problem, where we're getting compression ratios between about 1 and 5, which is essentially under what JPEG is really even capable of. We see JPEG starts above, a little bit above 5, maybe around 6 or 7. So we see that CAE is compressing the images, and we saw from our reconstructions that it is capable of compressing the image with low distortion. It's not quite on par with JPEG just yet. So just to highlight what I mean about the CAE being a bit sensitive to what is actually being compressed, this is just another image example here. So as I said, it's built with 32 by 32 images in mind, and large images are processed in patches. And an observer viewing them from a larger, from a large distance away, or whatever, this effect is sort of minimized. The distortion isn't as noticeable. But if we were to look at an actual 32 by 32 image, compress that, and see how we get with the reconstruction, well, what happens? So during the Winter Symposium, this was a user or an audience interaction portion, but I think I might be a bit short on time, so I'm just going to fly through this. But my question now is that this is a reconstructed image from CAE. Original dimensions are 32 by 32, and the question now is, what was the original image? So it's from the Linnaeus 32 by 32 data set. A few answers we got during the presentation were maybe it was a fly, maybe it was some sort of insect. But what is the image depicting? One that our supervisor said that definitely seems very plausible is a dog with a sausage in its mouth. But unfortunately, it's actually, although it's pixelated and maybe a bit difficult to see, it's actually a set of berries from the Linnaeus 32 berry category data set. So it's clear we can see that this, from the larger images, when we reconstruct them from patches, that distortion is sort of minimized. We're not seeing this, we're not seeing the dogs with the berries, so to speak, down on that sort of lower level. When you zoom out, it isn't as noticeable, these distortions. But when you actually look in, depending on the image, again, not all of the images from Linnaeus end up looking like dogs. A lot of them do end up still looking like their original image, but we do run into some issues like this, where they look like a dog with a bone or a sausage's mouth. And this artwork was done by Aoife, so thank you for that. So in conclusion, this project, we hope, has displayed the potential of learning-based methods for image compression. So we've achieved reasonable results with simple implementations. And as we said, our analysis was mainly based on MSE. Other metrics like SSIM would be more suitable for measuring visual perception. And just in terms of, there's been work done previously using a compressive autoencoder with convolutional neural networks that show effective use, in fact, even improving on JPEG for image compression. And our implementation is capable of reasonable, and we'll call reasonable to be about a ratio of about 5 image compression. But we can see it is heavily influenced by the training and testing sets that are used. And a focus on maybe more on compression in a more specialized area might be where CAE could improve on JPEG. So for the ERS scan, in most applications, the JPEG actually performs better than the GAN, but there are instances where the GAN is on par with JPEG. And we can see that there was areas where it was capable of lower distortion, and in particular, extreme compression ratios greater than 100, which is something where JPEG struggles with. And just one final bit on future work. We mentioned using SSIM or other quality metrics to train our models, and using more specific or niche areas like cats or medical imaging for these pipelines to sort of outperform JPEG as a general, which is more of a general sort of image compression pipeline. And then would be a GAN CAE hybrid. So we'd essentially stick together the two pipelines and get a new sort of novel compression method. So what we would do is we have the GAN with its input image downsampling. Then we have our compressed image in the middle with our GAN super resolution and our output image at the end. But then what we do is we put the CAE in the middle. So what ends up happening is the image is downsampled, then it gets fed through the CAE encoder. That CAE compressed file is now the compressed representation of the whole thing. Then if we want to reconstruct it, that goes through the CAE decoder. Finally, the GAN super resolution decoder, and that gives us our final output image. So there's a few of our references. Of course, anyone can reach out to me if you want to read more about this or see any of our source code. But otherwise, thank you.
that was a great talk, Paddy. I'm just going to open up the floor now to see if there's any questions, and I see one there in chat, I think, from Cormac. Yeah, so Cormac here asks, thank you for the talk, Paddy. Can I ask, how do you change the level of compression achieved?
Yeah, so let me just get one of the plots here. Yeah, so I mentioned earlier in this slide here. So so each of the CAE, how it works is obviously we train the model a certain way. We train that with a particular latent representation size in mind. So just this example here of this base 3 number is 512 digits long. So that means that this model has a latent length of 512. So you can train the CAE to have any sort of latent length, usually working in powers of 2. So 512, 10, 20, 10, 24, going up like that. 512, it sort of depends on what your images are going in. But essentially, if you want to change the compression ratio or change how much compression is there, you just have to train a new model with a different latent length. And then essentially, you would have that model stored where you'd be like, okay, I need to compress an image to whatever this ratio is. Okay, I'll use that model. If I need to compress it to a different ratio, then I'll use this other model. So that's basically how we'd change the compression ratio.
Yeah, that's perfect, Paddy. Do we have any more questions there now? Well, I have one there at the moment, Paddy. So it seemed that JPEG sometimes removed some of the color channels and converted images to grayscale in some areas. I'm wondering, do you think using the ESRG GAN, and if the ESRG GAN and CAE reduced color to grayscale in some areas, you think that could enhance compression further?
Yeah, I'd say definitely. We mainly most of the images that we work with, I'm pretty sure all of them from the data set, we're all just full color images, and in general, any of our code, we'd just be assuming that they're all color images. But yeah, I think definitely using grayscale instead, you know, it would just reduce, even just say, if you thinking of how we process the images or how we just put an image in, you know, usually it's like a 4D tensor or whatever, with, you know, a layer for sort of each different color channel. Whereas like if you reduce that to just, you know, one, if it's a grayscale image, you know, getting rid of those two sort of layers, it would definitely both improve the speed in terms of what we could do in terms of actually training the model on that. But I think you could definitely get some extra sort of compression in there with the grayscale, like you say.
Thank you, Paddy. If we don't have any more questions, I think we'll wrap up the morning session now. I'll pass back to you, Heidi, if that's okay.
So thank you so much, everyone. I'm just going to stop recording now.